Hello students, welcome to APG Pathshala. I am Indira Ghosh from JNU. Today we are going to talk on the module which is Prediction of ADME Property and Toxicity under the paper Computational Biology. The learning objective here to introduce you what is ADME properties. Mostly the prediction how one can do some of these properties. ADME properties are absorption, distribution, metabolism, excretion and toxicity. I am actually introducing you about how this absorption, distribution, metabolism by system or human systems and excretion are taken care of. How the physiology of this, addition to how one can formulate models or in silico practices so that we can predict some of these. So here is the actual system which takes care, physiology system, and that is gastrointestinal tracts. Everything is done after the bioavailability, that is controlled how much the hepatic extraction, which we call first pass, and how much is the balance of absorption in the bloodstream. After from bloodstream when you get in, there is a fraction which actually escapes the first pass metabolism as it is looked picture. Now from there it starts the distribution which is controlled by the ability to cross the affinity versus crossing the membrane. So some part might be affinity to the membrane is high, then it will be stuck there, then it will not be distributed. However, those parts which is not so much, which is more solvable, they come out and they are distributed equally. In the center, you can see the two major things which is our organ, liver and kidney. These two has very good operation and direct connectivity to our prediction of absorption and distribution. So what we notice, the difference of this absorption ADME property basically is very much dependent aqueous solubility of the molecule, ability of the molecule to cross the membrane, and resistance to the efflux mechanism or metabolism. Hence, many methods have been suggested to the predictions of these topics. I would not go through much detail of every one of them. However, some of them I have shown in our module 2 and previous modules, module 10 and 9, where I have shown you how in silico method you can use for actually how the membrane binding or metabolism or in liver binding etc. can be taken care of. What it actually depicts is the process. That is where the drug discovery process, in drug discovery process, actually one has to do a pre-formulation and then the final clinical. Now, before reaching the pre-formulation, one should understand that what are the property of the lead-like molecule should possess. Now, earlier I have shown you by high throughput screening and docking modules, how you come to a conclusion of hits, that is, which is possibly go for lead. At the level of this particular modules, we are discussing how to convert from heat to lead. And just because the lead molecule should possess not only its binding efficiency to the target which it should work for, it should also be available in the place where it should go. Hence, some of the physicochemical property which has been discussed earlier by Lipinski rules and some of the pharmacokinetic properties which should be designed here should be appropriate so that the lead molecule can be identified properly, or from heats we can design the lead molecule. So what are the techniques used? Major techniques are QSR to enhance this from the successful heat goes to the lead and actually goes through the pre-formulation and optimization. Now Lipinski's rule of 5, as I have discussed in earlier module, contains the data-driven knowledge on what makes the compounds to become pharmacologically very attractive or drug-like properties. And what is that? That is that if we do not satisfy these 5 conditions, most often, at least the 4 of them which is shown here in the picture, at least then or the molecules will not have a good absorption, permeation, and also probably the metabolism or excretion. Now, these few criteria is not a mandatory that the compound becomes absorbed. However, if you do not take care of them in the initial stage, there are high chances that the compounds might not be really good for ADME properties. So, I like to emphasize here that this is not a property which is makes a compound to be supportive to a ADME property. However, it is the few quality if we take in the initial stage taken care of, then we don't lose the chemical synthesis, etc., and testing in the next stage. The Lipinski space, which I've discussed earlier, and its relevance. Now, as you see, it has rule of 5, and if you take all the available chemical molecules or chemical drugs databases, basically, these are the compounds from where the molecule drug designing is done. All of them, it looks like 80% of them have a good permeability by these 5 satisfactory conditions told by Lipinski. This implies that a membrane permeation is not be the very much discriminant. However, these drug-like and non-drug-like molecules, we require to have some kind of cutoff to be decided. Hence, from high-throughput library or hit library should be well inside the Lipinski space. Tells how you do design your informatics drug discovery from your hits library. And how you do it, the objective is you supply the information and optimal generation of new materials. The objective is to extract and optimize as best possible the compounds which can be having its measurable properties, and you can test those. Also, this should reduce the optimization cycle. That means many compounds which will not be appropriate, they should fall fast, so that then we don't have to redo the same molecules. Actually talks about what are the criteria we should start a lead library, not Lipinski's rule. However, the reason is heat compound should have starting library to become lead must have less amount of numbers satisfactory, because during the optimization, which I'll be talking about next, we should be putting these fragments onto it. We will add some more to the HTS hits. So it has come up with another paper publication that the cutoff between should be reduced between 150 to 400 instead of 500, and C log P should be having a little less than 4.5, because we are going to increase C log P. Okay, then number of rotors, that is how many, that should also be less, because we are going to add few fragments. Similarly, we should have a reduced number of rings and reduced number of donors and acceptors. Actually, how one used informatics to optimize this cycle. There are plenty of optimization methods available which is regularly used by chemical engineers, and any one of them can be even adapted here, but I will not go in the detail how to adapt it. Rather, I will request you to focus on what are the space we should use and what techniques may be like cross-validation to have the process done. Hence, what we require is activity and property space of those chemicals. We should also have predictive models built up by mathematical generated models, which I've discussed even earlier modules. And we should have a proper cross-validation done. These are most important for designing this space. Describes each of these called space. One is structure space. What is structure space? Your chemoinformatics modules will give you the idea about the structure phase. Here I just repeat the structural relationship between the structure, that is their 2D topology, their connectivity, etc., with the surface relationship or interaction relationship between pair of molecules is what you should make a form of matrix. Many of such matrices have been discussed even in earlier modules. Next is index space. Index space decides the biological relationship, and this index space are something like related to your descriptor space or parameter space, and this also connects the from structure to the index. As discussed earlier, here I show you in principle how one must look into the model build using structure properties, indexes, and activity or property space. So as you see on the dark red color, that contains all the structural spaces, and the blue and the common area should have, the bottom one is the index properties and the right hand side is the property based. Common between each of these circle has to be so good that we can pick up the compounds of those properties and build a model from these data sets and predict, and the compound must be following the lead activity which the model is supposed to be generating. This slide actually shows the difficulty of achieving such goal. The main important problem here is most of these properties are not unique like Vidya-mitra, not like the good peak like you have shown on the right hand side top. Properties are described here. The drug-like are the bottom lines, and then as you go higher and higher, you can see it. The in between, the lead-like properties or some other properties actually decreases the number possibilities. Not only that, there are few peaks which could never reach to the drug, actual drug. So this data or the plots actually came from the data has been collected over a period of many times on the WDI databases and gone through how these compounds have been derived from the medicinal chemist bench till the become the drug. This is the difficulty in designing and which will be discussed much more details in caveats of drug designing. Actually tells you how to generate from such a complicated space the real virtual chemical library, starting from virtual chemical library to go to drug-like library. So how it generates? You get a compound of state of virtual library and of course the chemical space. You can use all your target filters in the beginning to decide about this chemical space, how much you are going to utilize. However, after that, you must run the DMPK filters. That is what I said, ADME filters, which should have the properties to filter out the like Lipinski rules or drug-like filter, which you may have designed by yourself. I will talk about drug-like filter, how to build in the last part of my talk. However, you can use these filters and then convert this molecular table into lead-like, which you get, and drug-like, what you get. So last slide shows there might be a very small space available, only drug-like and lead-like common. So we must be very careful to pick up compounds which should cause or which should be lying or contained in this common subspace. And two methods, that is virtual screening of libraries as well as chemical space and lead-like filters, which is docked or docking system are used, are very efficiently used to reduce this predictivity, etc., and reduce the of hard work and many quite amount of cost is involved in the next level of testing, because next level of testing is the development from hits and lead-like molecule to actually go like a drug-like molecule. Addresses one unique method and one unique system, and that is toxicity, which I have not spoken at all. And I will come to definition of toxicity. One is chemistry-derived toxicity. What it shows here is a picture which tells all things are poison and nothing without poison can be possible, only depends on the dose. That is how much concentration one takes it. So even salt, fat, alcohol, aspirin, even the famous drugs, all of them follow the same. It is the dose dependency decides the toxicity with human being and other testing material. Now, in this level of research of toxicity, mostly the actual experiments are done on a full-fledged system, not on organ. Hence, there are much more cost and much more risk is associated. And if some extent one can predict toxicity, would have been great help to save many of these mouse, rat, hamster, guinea pig, and sometimes even dog and monkey testing. However, if you look at two tables shown on left-hand side and right-hand side, there are two very famous drugs. Actually, the left-hand side has tetrachlorodiabenzoxin, which is a very important drugs. But if you look at the LD50, that is the killing dose, almost the lethal dose, microgram per kg unit, you can very well see the mouse and rat are systematically different from hamster. So if you do the experiment on hamster, they can tolerate much higher level of toxic on that, whereas the rat cannot. And if you go down at a human, we have not been able to test because it is so low amount can kill a human being. We are not allowed to do the experiments in this case. Hence, the predictivity is the only way out of this type of toxicity. Take the right-hand side, which is a drug. Lysergic acid is the one famous drug or, opioid, which is very, very commonly used as a pethidine and other injections sometimes. Look at the table shown, not only the chemical structure. The table shows that man has very, very low tolerance, whereas mouse has almost 10 to the power 3 times more tolerance. So if the experiment has been done on mouse, it is not possible or feasibly actually the man's tolerance to be predicted here. So toxicity is one of the most complex system and complex understanding to predict. Hence, but if we can predict, many helps can be done, which has been attempted. This is the chemical idea or chemist idea of toxicity. Now I come to the biological idea. Shows you how the human polypharmacology, which very recently has been unfolded. 2006, this Nature Biotechnology paper actually started this work and many papers came out with it. Here, what they have done is in this whole work, and which shows you the toxicity measure by taking the data of thousands of chemicals and their testing done at the experimental level in the laboratory, that what are the lowest amount of IC50 they affect or inhibit that target. Hence, one side we have collected several enzyme and receptors molecules for their titles, and other side, each one of the chemicals tested here were tested at least more than 3 to 4 different kind of activity or different kind of targets. Once it is plotted with the nodes as it's shown, at least 2 proteins are called to be interacting by the chemical method if both bind 1 or more compounds within the defined difference. So at least 3 compounds should bind, and it has been taken like that. And if you plot this in a network system, very obvious what you look for is that a picture which is shown here by the picture is there is a blob in the center. What it tells you that many GPCR enzymes, phosphodiesterase or even the phosphatase and many others are actually interacting more than even 3. And that shows you the nodes at the center. So many chemical compounds are acting on different things. Different things means different targets. However, they have been designed for one. And if you look at a few more exceptions, which is in ion channels, the chemicals which has been designed for ion channels, or you look at some proteases, you will see they are on the periphery, which tells you that these molecules are, the targets are very unique and not every compound can interact. It is kind of genomically looking at the, which is called polypharmacology interaction map of the human system. However, this has been really mapped much better later. So, 2 things we have come to know. One is many compounds would bind to many targets. And this is not very uncommon, which will create toxicity. And please do note, all of them might be inhibitory compounds. So, the lower energy was, lower cutoff of inhibition was less than 1 or so, IC50, 1 micromolar or so. Many cases, even 10 nanomolars were restricted, and it has been found they do inhibit the other targets. So this is called in biochemistry cross-target interaction. This is one reason of toxicity, and earlier I showed you dose effect is another reason of toxicity. Actually showed you our group approach, and we participated with a large number of people. I'll show you last that effectively we come up with some kind of prediction model of toxicity. Many other people have also developed each toxicity model. What we have approached here is coming from the chemical process. So cluster these chemicals and find their most maximum common substructure. That is, which is the maximum structure which is responsible for toxic or non-toxic, and create the dictionary specific for them, point of toxicity. And then you use the fingerprint and other method and predict it by random forest method, one of the artificial intelligence method, and predict the model. However, this model has been generated, or the structural handling such a huge amount of library and handling the fragment has been done by a technique called API, which are in integrated system in the chemical structure predictions, etc. So I will demonstrate to you how this works, or what are the major component of this and how efficiently it works. Shows you what are the data set. This is a limited data set, and it has been much better expanded later on. However, this is a limited data set we took from DSS talks, which are available freely with a threshold value. Dose was TD50 values known to be, and this test has been done only on rat. Same site we have actually taken others also, like many of the other TD50, body weight, chronic other data. So we have model for many different kind of LD50s considered or TD50s considered. Our data set contained 451 molecules, and one of the topics of toxic range I show you by next few slides. The method is and model building. We use the machine learning random forest model, and this method is used very much with divided into training set and test set, like 321 chemicals were training and 130 in test set. And when we designed this, as I told you, we first extracted the scaffolds, that is the fragments or maximum common substructure. That approach gave us initially unique 7200, so 7,000 unique scaffolds. We built the trees with that and a huge number, and did the seed and did many kind of simulation with that, and increased the number of or filtered the trees, and used a threshold of 1 nanomolar dose, which is very conservative for prediction. Papers shown down, we followed the same techniques. Correspondingly shows two models. One is using exclusively all the 7,200 models, and you can see the models, how they are predicted for actual values, and training set and test set. You can see the differences. We can also see the toxic, which are actually predicted toxic, non-toxics, which is predicted toxic or something like false positives. And if you look at the test set, you will see these false positive and the negatives are very disproportionate. So prediction method of using the whole 72,000 scaffold yielded us very good toxic to toxic. However, it failed very badly from predicting the non-toxic set, which should be called non-toxic. So many of non-toxic actually came in the toxicity. This is the difficulty to predict most often the toxicity space. However, this is a very conservative model. It is preferable that we predict better toxic molecules as toxic. However, some errors in non-toxic molecules would not create much problem. The same data is repeated and shown by choosing most important only 40 scaffolds. That is, instead of using all the scaffolds, I shown you the first few important component provider, which is 40 scaffolds. Here also we see the same kind of differences, and here the non-toxic prediction seems to be little better than the earlier ones. So what we come to a point, different models gives you different kind of scaffolds, and your performance sometimes decreases when we reject the non-toxic or less amount of contributory. However, it is preferable that we include as much possible informations. So in prediction of this case, study shows us also that. But I would go to the showing you most important thing, and that is 40 important scaffolds. For a designer, these are most important things. So we have predicted these 40 important scaffolds should be used in the modeling and should be used as a filter to have a scaffold which may cause toxicity. Now the question is, how you know that they are really good? Okay, prediction, numbers, all are correct. Where are the experiments? Actually, what we did was, we took the experiments, and this is how I have displayed here. When you build the model, please follow these. And that is highly important to make any QSR, any type of QSR. And that is already known toxicophore, which has been identified experimentally, and potential toxicophores experimentally found out are published. So what we did is we started searching how many of our molecules contains it. And here I show a list of hits where we have all these top known toxicophores are occurring in our molecules and in our predicted molecules and in our scaffolds as well. So this proves the mechanistic view as well in this. In this particular exercise. In summary of this method which I have displayed, we must know this toxicophores, and more we know the small toxicophore, and which will become found to be responsible for toxicity, larger the size is, it will be more importantly, we can design compounds not having those toxicophores. We should also understand from this exercise, and which we saw, that it is possible actually that to suggest small MCSs, small fragments, so that medicinal chemists are very carefully design the compound when they complete the lead synthesis. Then the results also has shown, if you use instead of 7,200 scaffolds, I use a limited 40 scaffolds, the regression values becomes significantly lower. Hence, classification is not though affected, but regression value, losing the regression value is not advisable. So already what I have discussed earlier, and it is emphasizing that the number of scaffolds have a cumulative property, some kind of cumulative property. Hence, maybe two scaffolds together is making it even better contributed to toxic or untoxic. Also, this model tells you that there are uneven distribution of the toxicity data, which has been shown in the earlier histograms, which makes also impossible to make a very good regression model. This summary not only talks about toxicity and chemicals, it also depicts or elaborates to you how the QSR models can be developed and how they can be analyzed and what are their limitations. I must acknowledge the team which is shown in the next picture of 11 countries participated, including JNU for this particular work, and Seascape supported us with a personal salary, and it only tells you one thing: the drug designing is not a one-site work. It requires many experimental data generation as well as IT facilities and the pharmacological aspect. So this team worked under OpenTox, and it's available all this model under the present OpenTox, which has been cited on the website. Actually addresses the caveats in drug design, which is very important, and that's what the rest of my time I will devote onto describing this. It's highly important. So QSR and have the most difficult work is that we always use linear relationship between the biological activity or ADME property or solubility or any of such properties with the structures related descriptors. However, it has been well proven by experiments as well, and one of this very famous plot I show here you that log P, which is a very important function in deciding ADME property, is actually non-linear relation with descriptors. And the reason is given here. That is, it decreases its activity due to the limitation of solubility beyond certain amount, or due to the protein binding. If the molecule has a property of binding to protein, it will be binding to many bovine serum albumin. Many serum albumins are available in blood. It will stuck to it. So obviously, it will be estimated much higher than actual log P. Hence, it's not too good to use the linearity relation up to the mark, maybe the optimal log P values we can. Now, if you ask me why we should know this, because many a time medicinal chemists synthesize compounds which goes much above the log P values. We have to be very cautious to apply any of the earlier taught principles like Lipinski's rule or other methods or descriptor-based methods to come to a conclusion by using linear log P. This does have important effect when you discuss about compounds which neurotransmitter takes them in or blood-brain barrier stop them, etc., which has not been discussed at all in my modules. The next caveat, how one does the designing of any molecules. Most often what we do is we design the compound, make it highly potential, fantastic, good binding affinity as it's shown by 10 steps or 11 steps. And then after the 10th step, when the docking, etc. has been optimized best, then we start reducing, at the cost of reducing the binding efficiency, improve the x-axis, that is poor ADME properties. And then land up or expected to land up as a compound as number 23rd step, the compound should have quite near to the binding affinity molecule as well have a high ADME property. This is called traditional discovery approach, used to be all the time. And this is a paper which has been done by collecting the information with my friend Tudor or Priya and the Ghosh and Vishwanathan's book called Decker. And this is been changed totally after this Lipinski and lead and whatever earlier I discussed about in silico methods. In silico methods, on the other hand, uses different. Traditionally, one needs to see how it has been, which is depicted in my previous slide. However, if we take the literature-driven data and analyze everything, we should be able then to make compounds only start from a lowest possible toxicity and apply all the filters and make the compound to go absolutely top. That is, it should have the very good bio availability as well as very good docking or very good binding ability. This is where the part of good ADME property and with the best property of good binding capability, both curves can map each other. However, it's not necessary that we could have used only this ADME property 5 rules and log D7, etc. or solubility, which I have not discussed much in my modules, and achieve that. Simultaneously achieving all these steps is what should be the actually expected solution, which is shown in the next slide. So, ideal, the discovery should grow hand in hand. That means, as we make the compound to be better potent for biological targets, we should also properly estimate the ADME property. That will diminish the number of steps, and designing capability should be taking here the course so that optimizations, etc. can be done earlier in the phase than waiting for the later phase. Start showing you many other caveats. Not only that, depending on the learning lesson of the medical science and the medicinal chemist and pharmacologist, these few plots, which I will show one by one, talks about the many caveats in drug design. And this is for the future pharmacologists who would like to make in future pharmacokinetics optimization, etc. optimization should be taken care of. Number one is, yes, it is a huge pharmaceutical industry, requires huge amount of money. And in the initial stage of designing, the money is spent on many compounds and many molecules, and it is much less per, if you call it, how many NCA per year or caught. However, as the phase increases, which is shown in my right hand side on the top, you can see to release to the compounds one, you spend more money on less number of compounds. This is the most difficult part. Hence, if we can reduce this, at least first two steps or the bottom two steps, and reduce the money, it will be useful for designing compounds much faster and economically much better. Next, analysis shows us that major reasons for failure in anti-infectious drug development, which is the most danger coming and waiting on the road, and major designers should pay attention onto this. And that is, if you look at the major failure is in lack of efficacy, which is shown in dark blue, and also certain extent at least, which is shown in green color, and that is mostly in animal toxicity. One should have to pay these reasons very strongly. Efficacy and this animal toxicity should be taken care in the earliest possible way. If we expand this anti-infectious disease to all the drug development, has been this paper cited, has the plots are taken from there, you would see that the maximum discrepancy or maximum failure consists of lack of efficacy once more and pharmacokinetics. In case of, if we look at other drugs, or you include the infectious disease but the rest of the drugs. Hence, kinetics, pharmacokinetics and pharmacodynamics to evolve properly, many softwares are developed to take care of that lacunae. However, lack of efficacy is a complex system. Many of the methods in biophysiologically and pharmacologically is involved to do this. What makes drug-like? A good group of people in Kubini and as well as Ghosh and Crippen data quite early has taken that whole database of the compounds which already been known in available chemical database. That means all the chemical from where you design your new chemical, they took that. They also took a set of compounds, so similarly the distributed compounds which was taken from already known dictionary of World Drug Dictionary, that is already known World Drug Index. And what they did is they filter out the duplicates between these, and they filter out the reactive compounds which is not going to be become drug, and then try to extract by artificial neural net characterization what are the property of drug-like. On the first plot on the top shows they got wonderful result, and it is expected result. So if you look at the drug score, the most of the ACD compounds which is not supposed to be drug, they are really lying within less than 0.02 scores. And when you look at the WDI, that is the most of the compound which is in the WDI, and test sets are dashed and training sets both coincide quite well. WDIs, yes, more and major number of compounds belong to WDI are in higher than 0.06 and 0.8. And it's very appropriate to see that. And it's very nice to know that really the drug-like property module which has been developed by them is working quite well. However, there are few troubles. And when they choose 100 top-selling compounds, which is shown in the bottom picture, what they found, which is shown by red color, many compound you can see at least percentage is almost 10% to 20% compounds are lying lower drug score. However, majority compounds are in the higher drug score. So there is a opportunity of developing. This is a wonderful model. One more demonstration how to build this model to relate the performance of drug-like or non-drug-like. This shows you the potential to come up in future for many large data analysis and developing model which can predict. However, here is the shocks to get. Many cases, already well-known fantastic good drugs like acetyl salicylic acid, which is aspirin, score is 0.30, which is below. Even diclofenac, which is we take it for our difficulties at night when you have cold and cough, it also has very low amount of scores, that is 0.40. So still we don't know, as it shows, it doesn't really map all the practical drug scores from the model build described to you. Shows you the difficulties and the future inventions or discoveries are waiting for. One of the most important thing to learn lesson from a very old person called Voltaire, who was one of the greatest philosopher of early centuries of 15th century or 16th century. He wrote this: doctors pour drugs of which they know little to cure a disease of which they know less into a human being of whom they know nothing. Seems to be still we are following the same process, but genomic systems and genome finding as well as understanding of SNP or nucleotide polymorphism or missing mutant or available mutants in the whole billions of people has given us a new opportunity to look at what are the human beings where we are pouring the drug. And that's why future generation should be looked into the in silico biology on pharmacogenomics. And that would bring us to understand the drug targets, how they are genotyped. Like every drug target may not be equally expressed, equally available for every drug we monitor. Hence, the dosage may be different. Also, depending on that, a personalized medicine which is coming up, new opportunities will be coming up. Also, every individual dose variance can be monitored. Also, the toxic effect and drug-drug interactions, these are major cause of even failure of drug in human beings, can be taken care even before treating the patients. So these are the hopes of future. How much we should trust about the predictions. So before I close, I would like you to bring to actual or factual world that all these predictions are so far I have stated and very convictionally I have persuaded and I ask you to do also use it. Predictions are predictions, and it's been talked by Niels Bohr as well as Mark Twain in two different ways. But summary of the statement is predictions is very difficult when you do about the future. However, that's what we are attempting to do. Here are examples. Most important, patent office in 1899, that is last 2 centuries back, told, oh, everything is already invented. We don't need any more patent. I don't need to tell you how big false that statement is. Chairman of IBM told, I think there is maybe 4 or 5 computers in the world. He told it in 1943. Then some of the people said, maybe it will determine the IBM's, the machines will be very small and they will have 1.5 tons. Nowadays, we have the more powerful machines or computers on our wristwatch. And the best one probably we should talk about, Bill Gates told in 1981. From Microsoft that hardly 640k kilobytes is RAM is necessary, and that should be sufficient, which is predictions done by drug industry and how bad they have been turned out to be wrong. There was the idea that cyclophorin is just too few organ transplantation. There is nothing market, but that is one of the $1 billion market present conditions because we need organ transplantation for so many diseases now. Breakthrough in medicine is available and that's why it is really wonderful. Cimetidine, which is a gastric drug, duodenal ulcer, claimed to be treated only by surgery. So there is no market for it. That's what's told. However, now $1 billion market for only for cimetidine. So then omeprazole also suffered the same way in the beginning that it will dry out. Nobody would use it. However, this also seems to be $1 billion market. Before I close, I would like to go to the final summary. I like to emphasize that we must remember predictions done quite way wide, quite looking at the present situation. So do the predictions are done by lot of validation supported and in medical science and medicinal chemistry as well as in drug designing. However, they are always been risky just because there are many biological components and effects which we are not clear even today. Hence, one has to remember the predictions are predictions, need to be verified really. Hence, one has to remember the predictions are predictions, need to be verified really by experiments. Final summary actually discusses here is that there are many data driven approaches, and especially employed in the case of this ADME property or ADMET even to be predicted. The main reason is this type of methods have high implicability in the case where actual all the biological and pharmacological functions are not understood yet, and that's why we rest on more of data driven methods than actual methods in previous cases like docking and other cases we have applied. Also, there are risks of this method. I have also summarized two different type of AI approach. One to handle the toxicity, another to find out drug-like molecule or pattern of drug-like molecules. But this data justification, that is how much data is available, is most crucial roles to play in the finally. Hence, data insufficiency also along with coverage of descriptor space, that is the descriptors calculation may give you lot of numbers, lot of parameters. However, the property which you are looking for may not be sufficiently covered by the descriptors. So these two insufficiency, one is not available full data coverage for the space, neither the coverage for descriptor, makes it the hardest problem in the drug designing work research. Last picture I would like you to remind, which has been taken from current science, that is the anchor for all data driven research. You require actually not only pile up the data but to analyze them as soon as possible. Thank you very much.